All right. So my name is Elizabeth.
I'm Dan Guinto.
All right, and we're going to talk about CRISPR today, and we came up with a fancy title of the CRISPR application for conservation. That's not a grammatical error of CRISPR. It's supposed to be CRISPR. CRISPR stands for clustered what that says, interspace short palindromic repeats. So we're going to keep that CRISPR, if that's okay with everybody.
So just a little basic background is CRISPR was discovered in the late 80s accidentally as like an immunoresponse of bacteria to virophages. And then more recently, they've discovered other applications for it in gene editing.
So instead of us kind of ranting about it for a couple minutes, we're going to take you back to biology class and see a short video.
Protein called Cas9 that functions like a molecular scalpel for DNA. The CRISPR-Cas9 system has an amazing ability to recognize a particular DNA sequence in a cell that may be malfunctioning and then disable it by cutting the DNA. We call this gene editing, and we can use it to disable or repair a mutated part of the gene which may be causing disease. For Cas9 to find the malfunctioning DNA, we attach it to an RNA sequence that matches the DNA sequence we want to edit. Then we put this RNA-Cas9 combination into the cell. It finds the mutant DNA and uses a chemical reaction to cut the DNA strand right at the spot where it's malfunctioning. After that, we can sometimes insert the correct version of the gene for the cell to work properly again. It's a very exciting technology that's going to do a lot of good in human society and for human health.
So our first application is to use a multifaceted approach of CRISPR to tackle one of the big conservation problems. Like the introduction last night said, when it comes to conservation, we are essentially losing the battle. And one of the big things that is happening is the amphibian extinction event that's going on in Central and South America with the chytrid fungus. So scientists have found some genetic resistance with MHCs, multihistone compatibility complexes, in the genes of some frog species, but not all of them. So basically, if we could use the CRISPR-Cas9 to locate that sequence on those frogs that have that resistance, and then introduce it into the ones that don't have it to try to give them a genetic advantage against the chytrid fungus. And then at the same time, bullfrogs and African clawed frogs have bacteria on their skin that show similar resistance to chytrid, and for that reason they've actually been spreading the fungus. So we thought if we could do the same thing with that bacteria and locate those same genes and then transfer them into the bacteria on the skin of other frogs, we could also make a bacterial community on their skin to also combat that chytrid fungus.
So clearly this is supposed to be implemented for fixing genomic structures, making them stronger, making specimens healthier. But I think there's an untouched approach which would be advantageous for invasive species, which is possibly making genomic structures weaker. By doing that, you can expose them to parasites that they might otherwise be resistant to, different diseases they might otherwise be resistant to. So an example of an invasive species that I think we're all aware of is feral cats. It's a global phenomena. It's not just in the United States, it's in the UK, and it's a big problem in Australia as well. And there's a lot of efforts going towards culling feral cats in Australia. I don't know if you guys have seen on the news, but a few months ago, someone announced that they were going to be culling 2 million cats. And, there's an uproar of, the ethics behind that. But in Australia, they are established as an ecological process that's harming wildlife there. So they're directly responsible for the extinction of 27 species there. So that's a great number of species, and Australia is unique in the fact that it doesn't have a native feline species there. So this would be the only feline species. So if we were to use CRISPR to make them genetically weaker to a specific virus, such as FIV, which is feline HIV, and infiltrate with that via fleas, then you have a way to specifically eradicate cats without affecting any other wildlife present.
And we thought of another similar multifaceted approach where at the same time we could also go in and experiment with using sex-linked lethal alleles to kind of throw off the sex ratio and kind of just limit the number of cats that will be being reproduced each year. We're also introducing maybe different autosomal heritable genes that will make them more susceptible to those viruses. And because CRISPR is getting cheaper, you could even buy basic sets for $100 to $400 online and kind of just play around with it yourself. So it's becoming easier and quicker to do. And a lot of people have focused on kind of capturing feral cats and spading and neutering them and then reintroducing them into the population. So we thought if we could just take in a large enough population and kind of just mess around with their genes so they're carriers for lethal alleles or for genetic susceptibility, and then release them back out into the wild and let their evolution essentially work against themselves to reduce their own population size. And we picked feral cats as an example, but it shows promise in other species, other invasive species, as kind of like a sixth tool to Leopold's wildlife management in reducing and eliminating invasive species.
Thank you. Any questions? Sorry, didn't mean to cut off the applause. All right, wait, one quick question and we're going to take a picture. If you can just press and hold on the microphone.
So I'm not sure how valid this is, but so if you're going to kind of gene edit animals, you're probably only going to be able to do that for a small section of the animals. And if they're genetically weaker, won't they just die off and the other section just survives? Like, how do you make sure this affects a large enough population of the animal?
So that's why we would have to ensure a large enough initial population, and we would make it sex-linked lethal recessive. So essentially, it would kill off a fourth of the, assuming simple Mendelian genetics of the sex-linked trait, it would kill off a fourth of the next generation and the successive generation, but it would also continue to carry that lethal allele on the females. So any male that would get the little X allele would essentially die because it doesn't have the other X dominant allele.
Oh, Alex, real quick.
So this idea of probiotics, I think, is a good one, but the challenge is how do you actually change the microbiome of all frogs? How would you actually distribute the probiotic on every frog?
So that's where we would have to take the bacteria and see if we could actually get it into the bacteria. And then if we could do that and allow the bacteria to proliferate, and if it would be a heritable sequence of that genome that it could pass it on to other bacteria. And then it would have to take some work to get it to spread actually on their skin and see if it would actually take hold on their skin. So.
Great. Thank you, guys. If you can take a quick picture.